Hello everyone, and thank you for the organizers for inviting me. Sorry about the slides, it's a bit, so I'll have to walk through it. So I am, as Elsa said, I'm a biology scientist working at the Polo Gorman with Alison Mickey and Tessa Holyoke. So I'm going to show you a project is slightly overlapping with what Elsa is doing, but is looking at slightly different questions. Not much about migration, as cell moving around the body in and out of lymphoid organs such as bone marrow and lymph nodes, but it's more about cell survival, so killing the cells. But mainly, one of the main things is the proliferation, so uncontrolled proliferation of cells, which is one of the main characteristics of progressive CLL, so poor prognosis CLL. So as probably you've been already told, the microenvironment, so what's surrounding the CLL cells in the bone marrow, in lymphoid organs, we call them bone marrow and lymph nodes. It's a plethora of cells which are feeding survival, so protective signals to CLL cells depicted here. I'm sorry about these cells is called nurse-like cell is off the screen, and you have T cells. So basically some of the immune system cells are providing constant proliferation signals and survival signals, and we call them proliferation centers in lymph nodes and bone marrow. So these are protective signals that have to be taken into consideration when we screen new drugs and find new targets. And so the main two things that we study in our lab are a signal called CD40 ligand IL-4, which is basically an interaction between two different cells. Physiologically, it is an interaction between B cells and T cells. B cells being the CLL cell, in this case for patient, and T cell, immune system T cells, which provides survival signal. Also already been touched upon the BCR signal, which has been exploited by drugs like ibrutinib. And both of these signals plus more, but for time constraints, I'm just going to focus on these two. We need to take into consideration these things when we are trying to find drugs that in combination or as a single agent will grant a cure for CLL. So what I'm doing is, as I said, slightly overlapping with Elsa. But I'm more focused on this drug on the proliferation side. And again, as Elsa well explained earlier on, there are two complexes in which this protein called mTOR works, mTORC1 and mTORC2. And they have different functions. You have mainly cell growth and protein synthesis. So is this complex one controls the machinery that makes proteins. And mTORC2 through a protein called, activating a protein called AKT, mainly activates processes to increase survival and perhaps resistance to drugs. So two crucial complexes for CLL survival and cancer in general, not just CLL. So as Elsa shown before this slide, there are several drugs that are trying to come in the market for patients, also in the lab as a basic research tools. And we are focusing on this side and trying to disrupt this negative feedback loop, which Elsa was talking about earlier, using this AZD8255, which inhibits both complexes, not only mTORC1 like rapamycin does. And it is in the context of the microenvironment I was mentioning before, and SDF1, as Elsa has been studying mainly, and BCR and CD40. So all the three main signaling. So you can just probably see it. So the first, this is in blue is AZD versus in red rapamycin after treatment of CLL cells from patients like you. So there is a little bit of an effect, not a great effect when you bunch all the samples together. When you increase the dose, there is not much effect, the higher the concentration. But when you start to split groups in terms of, in this case, cytogenetics, so if you're a normal cytogenetic, or you heard 11q or 17p deleted, the higher here the dots, which are some individual samples, the more sensitive they are to the drugs. So this slide is showing that AZD8255 is able to kill better the patients' cells, which are from patients with poorer prognosis as 11q17p, whereas rapamycin doesn't. It doesn't discriminate between these three groups, which is great news for an mTOR 1 and 2 type of inhibitor. This is just to show you what we do in the lab as a hands-on data. The dark bands are basically proteins detected by Western blot. We call it analysis. And when you see a band, it means the band is the protein is highly expressed. And here you have a list of proteins. The key ones I want to draw your attention is the AKT. I mean, there is obviously AKT word missing, and this is the active version of AKT, and this is the total version of AKT. You can see just only with AZD present. I don't know if you can see the plus. So only with plus AZD, you have a total knockdown of this protein, which is one of the key substrate of mTOR2 and one of the key proteins that drives proliferation, mainly survival. So we're quite happy with this, and it has many of these drugs. These are precise inhibitors. They have some off-site effects, off-site targets, but they're much cleaner than your standard chemotherapy. This is just an example, a schematic example stolen from Alice from McCaig, a slide that, just to summarize what we do in the lab. A few years ago, we just used to screen drugs on Petri dishes and just a culture medium, so liquid medium and nothing else, which is not very representative of any type of tissue or organ in the body. Then we started to use some extra cells, so we call it eco-culture. So we culture the CLL cells with stroma cells, which mimics the blood or the CD4 T, as I talked before, modeling the tissue compartments such as lymph nodes, bone marrow, where all those signals of survival and proliferation occur. So, and you can see here, again, stolen from Alison, during her PhD, she proved clearly that in plastic, which call it, this model, we call it plastic, you see a nice effect. So if you have the green, I would say green band goes down, means the cells are dying more readily with the azacitidine, in this case, in plastic. So you would think this drug is quite efficacious. But when you add the co-culture into equation, so when you mimic the lymph nodes of bone marrow, you have total elimination of the effect. But there is still hope, because there are some drugs which obviously are missing here, it's like CR8, a drug I was working on when I began to work with Alison Mickey, which is an inhibitor, inhibits specific protein that controls cell cycle. So that mechanism that makes the cells ready to divide and multiply. So a key mechanism. And you can see, although it's been reduced, you can see still an effect on cell viability, we call it. So if you have reduction, so you have more cell kill of the tumor CLL cells. So I think this is essential in order to test new drugs and combination of all the new drugs as well. In fact, in this context, now talking about CD40 ligand stimulation, same graph as before, but showing that basically that effect, although small, is completely lost when we add the CD40 ligand co-culture and rapamycin as well. Actually, the viability, so the cells are actually a bit happier with a little bit of drug, which is a bit worrying. But again, looking at the cell signal, so what's happening in the cells, again, the key protein here goes down slightly less than the plastic data was showing you, but still the targets are being hit. So the drugs are working well, but the survival signals from the culture are overcoming the death signals. And we perhaps think this is because of this little protein here, although you're missing the name, which is called MCL1, which is a key protein for survival in CLL. It's not really affected, is it, by AZD. So the middle band, this protein is affected by the drug, this protein is not, is completely unchanged. And we think this is one of the crucial reasons why AZD-type it doesn't kill CLL cells in this microenvironment model. But fear not, because we are also talking about proliferation, which is as important, if not more important, than killing the cells. We want to stop them from multiplying uncontrollably, so therefore they can't develop chromosomal aberrations, making them resistant to drugs. So what we do in the lab, one of the other things we do in the lab is labeling the cells. So we load them with a fluorescent dye called CFSE, which basically can track cell proliferation, and we can measure that in one of the machinery we are lucky to have in the lab is called flow cytometry, or FACS. So what we do is we label them, then we co-culture them as we did before with the CD40 ligand with different interleukins mimicking the microenvironment, so whatever happens in the lymph nodes or bone marrow. We leave them, then we treat them, and then we leave them again, looking after them, changing the drug and changing the medium just to make them as happy as they can be. And then early after a week, or a mid-stage after a week and a half, or 2 weeks in the late stage of co-culture, the cells will start to proliferate in this model, start to multiply. And then there are all sorts of things we can do through this flow cytometry machine. So one of the things we can do, we can trace exactly, this is, I'm sorry about the left-hand side, this is the intensity level of this dye, the CFSE, which allows you to track the cell's division. And without going into details, basically every time a cell divides, the dye will dilute by half in the daughter cells. So by division, each division peak will be dimmer than the undivided population, the undivided pool of cells. So the first peak here of this histogram, of all three histograms, is the cells that haven't divided so far. And then the second peak is the cells that have divided once at least, or once. And then you have smaller and smaller peaks and dimmer, so with less dye inside, which have divided several times. And with this, we can calculate how proliferation or cell division is hit by these drugs, several drugs. And in this case, we are looking at numbers instead of graphs, but cutting a long story short, here you can see that the AZD and somehow also rapamycin have a dramatic effect, dramatic effect on proliferation. And in this case, this value, the higher the value, the lower proliferation, the less proliferative the cells are. So it's just basically, this is just looking at the intensity level of the CFSE. So the higher the intensity, the less cells have been proliferating. And you can see here a significant increase. So definitely AZD, A255, to a certain extent rapamycin, inhibit proliferation, which is a crucial mechanism for CLL disease. So the other side was BCR stimulation. Without going too much detail, there is an effect. So again, looking at cell death or cell viability, so the other way around. So if the bar goes down, the cells are more affected by the drug. If it goes up, the cells are happier. And you can see this is cells unstimulated, so just the plastic. And this is with Fab, which is a fragment of an antibody which stimulates, engage B-cell receptor or BCR. And you can see that with stimulation, cells are happier, but then when you add the drugs, especially with AZD, cells are killed off. And again, this is just to show you hands-on data. Phospho-AKT, this crucial protein for survival, is affected only mainly by AZD8055. And I want to draw attention on the MCL1, which is the other protein involved in survival, is downregulated in or lower levels in the AZD group. So that's probably one of the mechanisms why cells with this type of stimulation are still sensitive. So it depends on what stimuli you give them. It depends on what the microenvironment they encounter. They will have a different response to the drug. So it's one of the things that we need to take into consideration. As Elsa mentioned, we also have animal models which mimics the disease. In this case, we have an animal model mimics the progressive, very aggressive disease. In fact, here you can see this is just number of cells we inject, number of soon-to-be CLL, mouse CLL cells. When we inject the mice, obviously, this is survival curve in days. Even the lowest amount of cells, 100,000 cells, will have a progressive disease. And obviously, if you add more, it's very, very, very quick. And we also have other molecular data suggesting that it's a very good match, not a perfect match because it's still a model of progressive CLL, which is what the current clinicians and scientists are focusing on because it's the group where needs the most effective therapeutics. So going back there, so we developed this model, and we use AZD8255 to see if, sorry about this, works. And here, just to show in the AZD, so the top 3 and the bottom 3. The top 3 are from one mouse that has been treated with just the vehicle, so basically control, no drug whatsoever, just the solution where the drug is kept. And this is with AZD. Without me telling you what it is, this is basically the CLL cancer in the quadrant. This is we call a dot plot, and every single dot is a cell from that specific tube. And these are markers for highlighting the CLL-like type of cancer. And you can see that with AZD is nearly cleared from the mouse. Here, just some numbers. You don't want to focus too much on this, but just to show you the percentage of CLL cells is dramatically going down. This is, each dot is a mouse, so is at least n equals 4, if not more, of mice. And then you can see the effect here compared to rapamycin. Rapamycin still has an effect, but it's not what we call a statistical significant yet. So that's the main difference between the two drugs. One last thing before I finish is about this. We were talking about for ages about this protein called AKT, and when it's switched on, it's switched off. One of the main events to switch on as a switch, basically, is phosphorylation. And this is what it says here as a P. A green P is an activating phosphorylation, so let's switch on. And as I thought before, mTOR2 switches on AKT. But these proteins, which are in this context are tumor suppressor, are FOXOS, are transcription factors. So basically, are proteins that regulate the gene expression. So very important proteins, especially in B cells. AKT phosphorylates, an active AKT phosphorylates FOXO, but then this is a switch off phosphorylation. That's why it's in red. So it's like a red light, stop. So FOXO can't work anymore, and it can't do its job in the nucleus where the DNA is, so for gene expression. And it stays in the cytoplasm, probably is overexpressed in the cytoplasm, as I will show you in a second. But what we think will happen, if we add a drug, we'll inhibit mTOR2, not with rapamycin, but with AZD, will inhibit this phosphorylation, which will obviously, AKT can't work anymore. Therefore, FOXO is free to go into the nucleus and work as a tumor suppressor, so will stop cell cycle. This is data from one of our students, an intercalated student, showing that the levels of, this is a technique called qPCR, looking at gene expression of RNA. And this is, each dot is an individual patient, blood from patients. You can see FOXO1 is high, we call it upregulated compared to normal healthy donor B cells. And also FOXO4, which is another family member of FOXOs. When we look at the biopsies in the lymph nodes, and this is thanks to collaboration with Mark Catherwood in Belfast, we can see that the indolent, so slow growing, like wait and see, you don't have much FOXO in the lymph node biopsies, whereas in the aggressive or progressive, as we call it here, you have upregulation. Here, just by, as a quantification, just a graph to see the difference between the 3 different types. But the story is not finished. So what we think is this, as I showed you before, so by inhibiting mTOR2, we reactivating FOXO somehow. And this is a piece of evidence suggesting that only with AZD, 8255, phospho-FOXO, the deactivating phosphorylation goes away. And one way of looking at, as Elsa was showing, is looking at fluorescence of nice fluorescence from CLL cells. So we're looking if FOXO1 with drugs goes inside the nucleus or not. And this is an image, but we've done more than one patient. We've done at least n equals 6. And this is the intensity of the ratio of the amount of FOXO in the nucleus versus the cytoplasm. So the inside, close to the DNA, or outside in the cytoplasm. And you can see with AZD, you have a significant effect compared to rapamycin and compared to no drug control. So we think, this is indirect evidence suggesting that AZD8255, so mTOR2 inhibition as an effect of this important tumor suppressor called FOXO1. So, in summary, I hope I explained a little bit about how mTOR1 and 2, so AZD85 blocks proliferation mainly in human CLL samples and in the mouse model, and how the poor prognostic patients are the more sensitive cells, are the more sensitive to AZD85 compared to the better prognosis ones. And this is reflected in our mouse model, which is always encouraging because we want to use this model for developing new targets. So it's good to see there is a parallel there. And also, we are showing you that this type of inhibitors, AZD85, is very good at reducing the amount of cancer in these mice affected by a CLL-like disease. And lastly, this kind of novel side of it, especially about FOXO1, which is a tumor suppressor in this context, we think is reactivated by the use of AZD85, which is another side of the goodness, I think, the utility of this type of inhibitors. And lastly, I'd like to thank everybody. As Elsa said before, I missed one center, but the Polo Gorman, where I work, the Bitson, where all the patients and the constant feedback from Mike Leach, and also McCaig, and our collaborators in Belfast, and of course the patients, because without you we cannot do this, and we are very lucky to be able to access to the samples. Thank you very much.